Hi guys, welcome to my YouTube channel. Don't forget to subscribe and share.
Assalamualaikum warahmatullahi wabarakatuh. So we're going to continue with the last topic in chapter 1, DNA and RNA molecule or nucleic acid. Okay, so now this is the last topic in chapter 1. The objective: describe the structure of nucleotide as the basic composition of nucleic acid. Okay, describe the structure of DNA based on the Watson and Crick model. State the types and function of RNA and state the differences of DNA and RNA. So nucleic acid is a polymer. So it's known as polynucleotide, consisting of many nucleotide monomer. So it means that the nucleic acid is polynucleotide and the monomer is nucleotide. So for nucleic acid, it has two types. Okay, first one is DNA, deoxyribonucleic acid, and another one is RNA, ribonucleic acid. So this is the basic structure of nucleotide that made up DNA and also RNA. It has phosphate group, pentose sugar, nitrogenous base. So again, this is basic structure of nucleotide. It has three components: pentose sugar, nitrogenous base, and phosphate group. Please remember this structure. And the pentose sugar is 2. Okay, this is different between DNA and RNA. Deoxyribose is a pentose sugar in DNA. So look at carbon number 2. Okay, for deoxyribose, the carbon number 2 has hydrogen in the position of downside. Then pentose sugar in RNA is ribose. So ribose, look at the carbon number 2, at the downside is hydroxyl group. So both are pentose sugar. They have 5 carbon, but deoxyribose at carbon number 2, the hydrogen is downside, or ribose is hydroxyl downside. Deoxyribose found in DNA, ribose in RNA. Organic base can be divided into 2. Pyrimidine is referred to organic base which have single ring. Purine is organic base which is double ring. So for pyrimidine has 3 types: cytosine, thymine, and uracil. Purine has adenine and guanine. So this is pyrimidine basic structure. Okay, look at here, the nitrogenous base in pyrimidine, okay, is single ring. So example is cytosine, thymine, and uracil. But for purine, the nitrogenous base is double ring. Example is adenine and guanine. Structure of nucleotide. Okay, so the phosphate group. Now we already discussed the pentose sugar and also nitrogenous base. So this is about the phosphate group. Okay, phosphate group bond the sugar to the next nucleotide by phosphodiester bond. So this is pentose sugar. The carbon number 1 will attach to the nitrogenous base, either pyrimidine or purine. And this is carbon number 2, 3, 4, and 5. So at carbon number 5, the phosphate group will attach. So phosphorate group is the group that involved in joining the nucleotide unit. So this is one nucleotide and this is another nucleotide. So look at here, carbon number 3 of the first nucleotide, 1, 2, 3, will attach to the phosphate group of the next nucleotide. So here 1, 2, 3, 4, 5. Carbon number 5 attached to phosphate group, and then the phosphate group attached to carbon number 3 of the next nucleotide. Okay, saya ulang. Phosphate group, okay, it will attach to carbon number 5 within the in the pentose sugar within the nucleotide. Okay, 1, 2, 3, 4, 5. So carbon number 5 attached with phosphate group, and this phosphate group will attach to carbon number 3 of the next nucleotide. So 1, 2, 3. So the attachment may form a bond called phosphodiester bond. Phosphodiester bond or linkage. Okay, here show the sugar phosphate backbone of DNA. This is nitrogenous base This is pentose sugar and this is phosphate group. Nucleotide attach with another nucleotide by forming covalent bond or phosphodiester bond. So again, the structure of nucleotide. So now let's proceed. The types of nucleic acid is DNA and RNA. Let's describe the structure of DNA according to Watson and Crick model. DNA is double-stranded polymer of nucleotide. The pentose sugar is deoxyribose. DNA will consist of 4 nitrogenous bases, which is adenine, thymine, guanine, and cytosine. So DNA is very long and thin molecule. The amount of adenine is similar to thymine. The amount of guanine is similar to cytosine because adenine will always and only pair with thymine. Same goes to guanine will only pair with cytosine. So DNA is in helical shape. So the helix shape is maintained by hydrogen bond. So this show the purine, which is the adenine and guanine with their pair. Adenine will pairing with thymine, guanine pairing with cytosine. So the pairing, how they pairing up? By forming hydrogen bond. So look at here, this is thymine, this is adenine. So they form a hydrogen bond. For thymine and adenine, they form 2 hydrogen bond. For cytosine and guanine, they form 3 hydrogen bond. In DNA, the polynucleotide arrange anti-parallel. Okay? The 2 strands of DNA double helix are found to be oriented in opposite direction. So look at this. The pentose sugar is facing upside, and then the phosphate group is facing upside with 5N. Okay, because this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. So the phosphate group attached to carbon number 5 and facing upside. So we call this as 5' N. And then at the bottom, this is carbon number 1, carbon number 2, carbon number 3. So here is N with carbon number 3 that facing downside. So this we call as 3' N. Compared to this polynucleotide, okay, this is carbon number 1, carbon number 2, carbon number 3. So means here, the carbon number 3 is facing upside. Here is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. So this is 5'. So the anti-parallel means the polynucleotide arranged in opposite direction. 5' to 3'. 3' to 5'. Again, it shows that the DNA is anti-parallel. Again, adenine and thymine, they will form 2 hydrogen bond. Guanine and cytosine will form 3 hydrogen bond. The number of adenine always similar with number of thymine. Same goes to guanine, or they will have same number as cytosine. Okay, settle about DNA. Then this is RNA. Compared to DNA, RNA is single polynucleotide. So the pentose sugar is ribose R. Function of RNA is to translate the genetic material stored in DNA into protein structure. So remember, function of RNA is to translate genetic material stored in DNA into protein structure. So the nitrogenous found in RNA is guanine, adenine, cytosine, and uracil. So means that in RNA, thymine is not found. In RNA, thymine is replaced by uracil. So for the pairing, guanine will pairing with cytosine, adenine will pairing with uracil. So RNA can be divided into 3 types: mRNA stand for messenger RNA, tRNA stand for transfer RNA, and rRNA, which is ribosomal RNA. So here it shows the RNA polynucleotide. Okay, the base consists of guanine, cytosine, uracil, and adenine. So RNA is single polynucleotide. Types of RNA. Okay, we have rRNA. The function is to combine with protein to form ribosome. Messenger RNA function is to provide the instruction for assembling amino acid into a polypeptide. Number 3, transfer RNA, which is tRNA. The function is to deliver amino acid to a ribosome for their addition into a growing polypeptide chain. So please remember the function of each type of RNA. You're going to learn in detail during chapter 6. Okay, the RNA actually involved in translation process. So this is the basic comparison between DNA and RNA. DNA is double strand. RNA is single strand. The pentose sugar in DNA is deoxyribose. In RNA, the pentose sugar is ribose. Nitrogenous base that found in DNA is adenine, guanine, cytosine, and thymine. But in RNA, the nitrogenous base is adenine, guanine, cytosine, and uracil. So this is the difference in terms of nitrogenous base in DNA and RNA. And this shows the difference in terms of pentose sugar. DNA consists of deoxyribose. RNA consists of ribose. So here show DNA is double strand and the shape is helical, but RNA is single strand. Okay, this is the base in RNA. So here, okay, the difference between DNA and RNA that we must remember. At least you remember 5. Okay, so DNA double strand, RNA single strand. DNA larger molecular mass because it is more longer compared to RNA. So RNA has smaller molecular mass. DNA, the pentose sugar, is deoxyribose. RNA is ribose. Organic bases or nitrogenous bases in DNA is adenine, thymine, cytosine, and guanine. But in RNA, the organic bases is adenine, uracil, cytosine, and guanine. DNA manufactured in nucleus, and it will be in the nucleus. But for RNA, it will be manufactured in nucleus, but found throughout the cell. DNA is chemically very stable, but RNA chemically unstable. DNA permanent existing, but for RNA is temporary existing. After translation process is done, RNA will disappear. And then DNA only has one basic form, but RNA has 3 basic forms, which is mRNA, tRNA, and RNA. So in DNA, the pairing is adenine with thymine, forming 2 hydrogen bond, and then the guanine with cytosine with forming 3 hydrogen bond. But for RNA, yes, they are single strand, but during translation process, the mRNA and the tRNA, they must pairing up, and they must be what we call compatible. Okay, so in this case, adenine will pairing with uracil because thymine is not found in RNA. Then guanine will pairing up with cytosine, and the ratio is varies. So that's all for 1.5. Thank you. See you in the next video. Bye.